decades. For our next panel, we're going to focus on heart failure and the therapeutic mandate. And leading this effort, we are fortunate to have Dr. Akshay Desai, who is an associate professor of medicine at Harvard Medical School and the director of heart failure management at the Brigham and Women's Hospital. Akshay splits his time between clinical care, in which he's renowned as an expert, and also involvement in clinical research, particularly clinical trials developing new therapeutic modalities for heart failure. So this is the other side of some of the device interventions we heard about, so we're really looking forward to this. Again, we have industry and academic leaders to discuss the heart failure therapeutic mandate. Thanks very much.
I think that song played at my wedding, so it's a different meeting than you thought you were signing up for. Yeah. So welcome. This is Heart Failure's Therapeutic Mandate, and I'm Akshay Desai. I'm, as indicated, a heart failure cardiologist at Brigham and Women's Hospital, and we have an excellent mix of device and pharmaceutical company industry representatives. To my left, Phil Adamson, Vice President and Medical Director from Abbott. To his left, Craig Basson, who's the Vice President and Global Translational Medicine Head from the Novartis Institute of BioMedical Research. Next down the line, Marc, Semigran, a former Mass General Hospital cardiologist and now Chief Medical Officer at Myokardia. And then to his left, at the end of the line, Ken Stein, Senior Vice President and Chief Medical Officer for Global Health Policy and Rhythm Management at Boston Scientific. The mandate for this session is really to talk about the therapeutic opportunity, obstacles to realization of that vision, and perhaps some of the pathways forward for academic industry collaboration in the heart failure space. And that's a broad agenda for a 50-minute interval, but it really derives from our recognition that heart failure is a growing therapeutic problem. About 6 million prevalent cases of heart failure, the leading cause of heart failure of hospitalization in the elderly or Medicare age population in the United States, high rates of readmission both at 30 days, which is our fiscal imperative, and beyond, and high rates of mortality after heart failure hospitalization despite considerable progress in pharmacologic and device therapy for this disease. And so in the next 45 minutes, our goal is to really outline the therapeutic opportunities, the research pathways needed to get there, and to talk a little bit about what obstacles we need to overcome to realize the vision for heart failure care in the next decade. And so I think with that broad background, what I'd like to do is to open up the panel discussion, because really this is to hear from you, and to talk a little bit about what you think are really the exciting developments and opportunities in the heart failure space. And maybe, Phil, you could start, and then we could walk down the line.
Well, thank you, Akshay, and thank you for the invitation to be here at this really impressive forum. I think one of the things that is on the forefront of my mind is the fact that those statistics that you just talked about are expected to double in the next 15 to 20 years. I think if we're having such a difficult time managing this disease now, what would it be like with twice as many patients? So there's a real urgency to find real solutions to help this disease become an outpatient process, to help this disease to be treated more efficiently as well as more effectively with new breakthrough drugs and devices and approaches to the management. I've been struck by the heterogeneity of how heart failure has been managed across the country, around the world. And I think bringing a more homogeneous approach towards outpatient management of this disease process, I think, is going to be very important in our innovative thought process and how we bring things to help our patients with heart failure.
Craig, you've thought a lot about personalization of medicine and the ways in which we can think about how to do that better. What are your thoughts about real opportunities, and what are you excited about in heart failure?
Thanks, Akshay. I think it's, first of all, let me say it's really a pleasure to be here, and thank you for coming. Thank all of you for listening and sharing your thoughts with us today and questions. I think Phil has done a great job at sketching out the impressive medical need. You know, we've had heart failure and dropsy for how many years and how many centuries now. We've moved from William Withering to Marc Pfeffer and ACE inhibitors, and yet we still have dropsy. And so it's a tremendous medical problem for us. But what's exciting to us now is the scientific opportunities, the new biology we're discovering. Heart failure in many ways has lagged behind the other areas of cardiovascular disease. Atherosclerosis, arrhythmias, we've had huge inroads in understanding the genetic causes, the molecular causes of those, and those have given rise to new targets that are targets that can be manipulated both by medical therapies and device therapies. Heart failure has been more intractable. We don't see a lot of large genetic studies yet. They're coming now to understand the clear genetic factors that are causing the heart failure. In part, that's because it's such a heterogeneous disease that we haven't really understood how to match those phenotypes with those genotypes, which is clearly the key. These things are upon us now. New studies are available to us. We're having new hope with translating the work that many of the people in this room have done in the past with Mendelian, rare Mendelian forms of cardiomyopathies to match those with the phenotypes and the genetics signatures in the general population. As we match those together, I think we have a new opportunity to personalize the medicine, to find new targets and pathways.
Marc, any further thoughts about the sort of the opportunities in heart failure?
Yes. Thanks, Akshay, and thanks for inviting me, and thanks to the organizers for inviting me as well. I certainly agree with what Craig said. We need to have cardiologists thinking more about how best to phenotype heart failure along the spectrum of possible etiologies, because that will allow us to deploy targeted therapies that really, I think, can get at the root cause. I mean, you know, our colleagues in oncology have been doing this for the last, decade or 8 to 10 years or so with a fair amount of success in not looking at all non-small cell lung cancer, for example, as identical, but looking more at what's going on at the genetic level and at the cellular level. We need to do that in heart failure as well. And then the second thing we need to learn is I have colleagues on the left and colleagues on the right who have devices that can be very effective at monitoring the physiology and the changes in physiology that are occurring in heart failure patients. We have to learn further how to utilize this information to vary therapies, to target therapies at physiology, whether it be a peripheral vascular issue, whether it be a central cardiac issue, right ventricle versus left ventricle. I think that we can target all of those separately, but we need to learn how to do it intelligently for individual patients.
And then Ken, maybe building on this device theme, I know that that's an area of great interest for you, and so perhaps some other thoughts from your perspective.
Yeah, I'm going to be disappointing and agree with everyone. I think the fundamental problem here, right, is that we treat congestive heart failure as though it's a complex disease. It's not a disease. It's a clinical syndrome that can reflect a lot of different diseases. I just actually flew back yesterday from Paris, where I was attending the European Society of Cardiology's heart failure meeting. It's the biggest heart failure meeting in the world. And it's distressing to me as a device guy. You look at the current guidelines for treating patients with heart failure in the ESC, and there's a single target dose of a variety of different medications. Well, that's crazy. Imagine if we lived in a world where the endocrinology guidelines called for a single dose of insulin for all diabetics, and if you weren't at that dose, you weren't treating your patients appropriately. I mean, that can't possibly be the right solution. The right solution has to be to titrate our therapies, whether they're medicines or whether they're drugs, to the physiologic effect that we desire according to a given patient's underlying physiology and disease substrate. And so to come back, you asked what's exciting. What's exciting now is that we have the growing opportunity to telemeter out various physiologic signals from patients' bodies, whether it's from implantable devices, whether it's from wearable devices, whether it's from permanent devices or devices that you use for a short period of time, and use that physiologic information to risk stratify patients, to understand patients' physiology better, and ultimately, I hope, to guide our therapeutic strategies.
Do you think as a group we have the frameworks we need to begin to make movement in those directions? And by that I mean, You talked a bit about the guidelines as an example and a fixed dose, but another way in which the guidelines might be a little arcane is that we've segmented heart failure into 3 basic groups, right? We have patients with acute heart failure, and we treat those as one entity. We have patients with low left ventricular ejection fraction heart failure. We treat those as a separate entity. And then we have this amorphous group of preserved EF heart failure patients, which are a 3rd group. And our guidelines then follow those columns. Is that a fine enough stratification of the population? And to your point, maybe there's a path forward here for more physiologic separation of those groups, which are maybe a bit old.
Well, I think you've hit it right on the head, Akshay. I think we're really just beginning, aren't we, to understand the morphologies and the phenotypes of heart failure. And if you add the ESC guideline recommendations of a mid-heft, now we have 4 groups.
Yeah.
But have really studied those with low ejection fraction. So we extrapolate many things to preserved ejection fraction patients, for example, in a group of patients for which there's been really no outcome-changing intervention. So I think that we have a lot of work to do, and understanding both the genetic basis of what's happening, but as Ken mentioned, to target individual pathophysiology, which I think has to be reiterated, that individual patients respond individually to drugs. We get target dosages from large populational trials, but when you're looking a patient in the face, that's one patient that needs to have their medications targeted for them. And then a pharmacogenetic approach may very well be the approach that will help us both transcend all the classifications of heart failure, but also help us understand the pathophysiology better.
Yeah, you know, I agree with you, Phil. I think if you even think about the categories you laid out there, and just start with reduced ejection fraction heart failure, right? We know that about somewhere in all of our trials, between half and 2/3 of those patients in a reduced ejection fraction population will have ischemic heart failure for their etiology. And then there's everyone else. And how can those 2 populations be the same if we think about them? And then what's in the everything else? That's easy to call it everything else, but what exactly is non-ischemic? And of course, preserved ejection fraction is even more complicated, right? If you look at our field, how many years did it take us to come up with the name preserved ejection fraction? And I think if we took a poll in this room, I bet we'd get at least as many definitions as there are rows in this room of what preserved ejection fraction heart failure is. So how can these be the same? So you have 2 choices if you want to take a molecular approach, this, and a medical therapeutic approach. You can try and sort this out, or you can take what we've historically done, which is try and look for final common pathways and target those pathways. And that's what most of our medicines, what our devices really do. The problem is you have to assume everything goes through the final common pathway, which sometimes works, but sometimes doesn't. And then the other issue is, of course, if you're in the final common pathway, those are really important pathways that are conserved in the heart, the myokardia, all the other layers of the heart, and everywhere else in the body. And so you have the risk-benefit ratios and the adverse effects on the patient that they can't tolerate many of the drugs we have anyway. So really, if you want to get at the bottom of this, you need to understand the different subsets here. You need to figure out how to identify which patients are in the subsets with some of the physiologic measures that Ken has been talking about. And then you need to have medicines or devices that target the particular pathways that are unique to those subsets. And again, that's what's exciting now, is we do start to have the populations that are amenable to that. In our company, for instance, as many of you know, we've run several large heart failure trials now. So all of a sudden now we have access in our research units to thousands and thousands of patients with multiple years of follow-up with phenotypes, blood samples, et cetera, to begin to correlate those 2. We're just at the beginning, but it's pretty exciting.
I certainly agree with both Craig and Phil. I mean, we need to particularly focus on the different genotypes and see what can that teach us about other types of phenotypes of heart failure. For example, the kids who have dilated cardiomyopathy, the young adults who have hypertrophic cardiomyopathy. What can that teach us about the pathways that, elderly people with HFpEF are developing. I think that when we've tried to do a good job at phenotyping our patients, for example, with the deployment of resynchronization therapy, that we've met with huge successes. But in the HFrEF population, honestly, we kind of pile one therapy on another therapy on another therapy. And, it's based on our knowledge of what worked in the past. But we have to step back and look, what are the true pathologic targets that we are approaching in an individual patient, and how can we best improve them with both medical or device therapy?
Yeah, and the one thing I'd say is we actually know a lot about this, and we just haven't brought the threads together. I was struck, we had the atrial fibrillation panel earlier, and as a recovering electrophysiologist, I paid a lot of attention to that. And one of the points that was well made there was how poorly we understand the physiology of atrial fibrillation, and how that limits our ability to improve outcomes. Within the heart failure space, we have a very good understanding, for the most part, of a lot of the pathophysiology that goes on. And so, we have known since the 18th century that abnormal heart sounds in S3 and S4 are bad prognostic markers in heart failure. We have known to listen to patients for RALs. We have known, to check whether patients are tachypneic or orthopneic or have PND. We know, you talk about the differentiation to HFrEF, HFpEF, but we've known for a long time that patients with ischemic cardiomyopathy behave differently from those with non-ischemic cardiomyopathy, and that those with large LV aneurysms are different from those with multi-infarct. But what we haven't been able to do yet has been to take what we know, and then translate it into what are the best therapeutic strategies for individual patients. And I think, again, the reason for optimism here is I think that the technology and the bioinformatics, the ability to do big data analyses, has got us to the point where we are on the cusp of being able to tie all those strands together.
So let me pick up on that point, this issue of the novel approaches that we have available to us and how it is that we might leverage them to make some headway here, right? Because I think you pointed out at the beginning that a lot of what we've done has been a little bit fortunate. We found final pathways that are relevant for a broad range of patients, the broad spectrum antibiotic, if you will, that works for everybody with a fever. But we don't have a way of defining the specific categories of illness. So how do we leverage some of these big data techniques or informatic techniques or genetic approaches to begin to get some leverage on how we can move forward with new areas for therapeutics?
So, I think that I think the big data techniques are here, and they give us a technology to manage these data. But they will continue to improve, a Moore's Law kind of thing, and we'll get faster and better. It wasn't that long ago, a couple of months ago, that I remember the fastest way to transfer a large data set from our Novartis campus in Cambridge over to the Broad Institute in Cambridge was to walk the hard disk across the street. So these will continue to get better. But I guess I would come back to that the real challenge is what's going into those data sets. It's the phenotypes, it's the genotypes, it's the proteomic data, it's the physiologic data that we need to collect with more and more precision. Because as we have these big data sets, the little bit of noise that we would have accepted before we had these data sets will overwhelm the system. So we need that precision. We need that in our trials. We need that in our follow-up in the clinics. It is, like all of these big data analyses, like genomics has been since long before I ever entered this space, it's been a garbage in, garbage out science, right? And so we need the precision and the inputs into those databases. And that needs to come from industry in how we do our trials, but it needs to come from all of the academic centers that are participating in our trials as well to provide that high quality data set. And how do we do that efficiently? There are a number of things. I know many of the people on this panel are concerned about how we collect those data in a cost-effective fashion, how we translate those eventually to registrable endpoints, of course, is a challenge as well. But that's where we need to grapple with. I think the technology is coming. It's here and keeps getting better.
But I always struggle a little bit with the fact that there is the way in which that data enters the system is so different depending on the institution. We have each institution as a separate electronic health record. Each institution collects data that's different on every individual patient. So is that to say that in order to move forward, we need to homogenize that in some way, or can we make progress as we are?
So Ken and I were in practice together at Cornell before we're in our current roles, and I'm sure Ken can tell you even more horror stories than I can about the early days of Epic at Cornell. I'm not sure if there's anyone from Epic in the audience, sorry. But as I'm starting to see some patients again at the Brigham, Callum McRae has forced me to sit through Epic training again. And it was painful. But it got a little better, I have to say. Not a lot, but a little bit. So, you can homogenize all these things. But as I sat through Epic training 2 weeks ago, and I started to ask questions, I realized again how woefully inadequate what the data that's being captured in Epic is, and how little it tells you, other than for billing purposes, anything about the patient. So it's still the data that's coming in, and how we homogenize it across systems, yeah, we can work on that. We need to get the right data in the system. It's the same thing in our trials, by the way. It's not just a clinical problem. We all have probably very different platforms that we use for ECRFs and all of those things in our trials. We can find ways to match those up, but it's the data that's going into the ECRF that's the key.
I'm going to take a little bit of a contrary point of view here. I think that one of the problem with the big data is still going to be who's entering the data. And that if we're really going to move towards precision medicine, we really have to face the patient in front of us and perhaps use imaging, perhaps use physiologic data, really to define, what are the best targets to go after in treating that person's heart failure. And I think we can learn a lot from adapting what we have already, as I think you said, Ken, devices that can download information, and novel ways of looking at imaging. I mean, CMR, for example, can provide us with important information about fibrosis, its extent. Is this a HFpEF patient who has an infiltrative myopathy? There's the new availability now of radionuclide scanning to exclude amyloid as a cause of HFpEF. And I think that we have to, still address the clinician who's facing the patient in front of them and give them the tools that they can use to deploy the best therapy for that patient. And it may well include genotypes. And I think that the challenges out there to cardiologists who've always been very good at adopting new techniques and new therapies to adopt an understanding of the genetic basis of many of our cardiomyopathies.
Yeah, there's data and there's data. And, probably beyond the scope of this to go anything further than agreeing with Craig about the deficiency of the current EHRs, EHR interoperability needs to happen, particularly needs to happen if we're really going to take full advantage from an innovation standpoint of learning what we can, right? Get into a era where we learn from every patient who's part of the healthcare system. On the other hand, and again, Phil and I may be a little spoiled on this because we come from an environment, right, where our implanted devices telemeter out data that automatically goes into the record, does not require any patient involvement, does not require any doctors inputting data, and it's completely objective. And it's one of the sort of interesting things. Again, I talked about sort of our multi-parameter heart failure diagnostic. And one of the most powerful discriminating factors for impending heart failure events turns out to be a device-detected S3, abnormal heart sound. That also turns out to be far more powerful than a physician-detected S3. And that's not just a comment on my skills with the stethoscope, right? It's a comment on the difference between having objective versus subjective matters. And so to the extent that the big data that we're looking at are objective and highly accurate and are automatic, right, it's much more powerful, I agree, than relying on some of the sort of cut and paste stuff that we know goes on in the EMR today.
Yeah, and I think one of the things that is possibly an obvious challenge is as sensor development, sensor platforms, as Ken has mentioned, as inputs come in that are more objective, one recognizes now how complex this syndrome really is and how for hundreds and maybe thousands of years we've recognized the syndrome, but now in the last maybe 5, we're really getting a handle on how do we actually monitor the patient. And oh, by the way, the tools we've used for that many years are not that good. But the problem, if we want to be pragmatic, is not that we don't have a genotype, it's not that we don't have a phenotype. It is that we consider this syndrome as easily treated. The cardiologist many times is not even involved in the longitudinal management of the patient with heart failure. That's relegated to a family physician or internist who can become quite skilled at it, but the specialist many times is not even involved in the patient's care when you get out of an academic center and in the real world. So the patients that actually drive the statistics we're trying to change, like Akshay mentioned, the 25% readmission rate, the 50% readmission rate at 6 months, many of those patients are being cared for by someone who has 5 minutes to see a patient. And I don't know about you, but when I saw patients, it took me 15 minutes just to get through their medication list. And so it's hard to take care of this disease thoughtfully and bring new innovations, as we've seen with new major breakthroughs in technology and in devices and drugs lately. It's hard to bring that innovation to actually the patient. So I think that one of the things that we have is a culture, I think, a biosystem of care for heart failure that is slowly evolving to recognize the specialist and recognize that the specialist shouldn't just be involved in the very end stage of the end stage, this natural end point of cardiovascular disease, but should be involved earlier on to deploy what we have, including CRT. I heard a statistic from Europe last week that 50% of people who have a clear indication, class 1A, indication for cardiac resynchronization therapy receive the therapy that's been approved for 16 years. So where does the biosystem that actually interfaces with the patient change when we're all trying to figure out how to punch the right buttons on Epic? I think that's one of the major issues in a therapeutic sort of mandate, is to deliver the right kind of care to the patient.
I also think that another potential target that we haven't looked at enough is stressing the system and having the patients exercise. I learned long ago from the endocrinologists that you can bring out the abnormalities in the system and hopefully bring them out earlier than when they're manifest as advanced heart failure by stressing the system. And in the world of endocrine, it was, whether to give an oral glucose load or give an ACTH injection. I think in the world of heart failure, it's asking the patient either with an implantable monitor to walk across the room or walk up a flight of stairs or doing a formal metabolic exercise test where we can really learn to differentiate. Is this a central cardiac problem? Is this a ventricular vascular uncoupling problem? Is this a peripheral problem? And I think that we haven't used that enough to help define the varying phenotypes of heart failure and to target appropriate interventions.
So I think we've talked a lot about the breadth of this disease and the ways in which we might take exercise, device-derived data, genotypic data, and other things to begin to describe more discrete phenotypes that hopefully will inform past the treatment. So then the question is that we start to define diseases that are much more discrete. How do we make progress then in defining new therapeutics? So I wanted to think a little bit about our clinical trial paradigm. Because as the populations that we're studying become smaller, then the ability to do large-scale clinical trials that are outcome-driven becomes a little bit more challenging. So as companies in different spaces related to heart failure think about this, how do you think about new therapy development in this space, in this evolving space?
Well, I think about that a lot, actually. And it certainly is a challenging space at the moment in the United States and around the world. I think it's this complex interaction between regulatory, payer coverage, reimbursement processes, and the independence that many times now we see those organizations have that really create a de facto multiple layered regulatory system. it becomes a real challenge to timely deliver an innovation that by the time it gets to the patient is still an innovation. And so I think we're getting better on some fronts, and we're not getting better on other fronts in the United States. Around the world, I think, we're seeing evolution of acceptance of technology sooner in some places and yet not in others. So this, as financial constraints, I think, hit systems and the realization of how many places are really broke, I think it's tough to innovate when the system refuses to recognize the innovation and cover it and pay for it. Because that's the only way that these kinds of studies can be done, is if they will pay off.
So I think that there are two ways to go about this. Again, is that the problem is exactly as Phil lays out. For a drug like Entresto, to bring that forward, to fully understand what its effects are in different forms of heart failure and heart failure patients requires the commitment of 20 to 30,000 patients followed for up to 5 years each. That's a huge commitment, not just in cost, but in patient effort and time dedication by the patients, physicians, et cetera. It's just not practical to do that for many therapies out there on a routine basis, obviously. So we can try and deal with the reimbursement side on one end to make that more affordable, and there are obviously a lot of discussions that go on about that. Or we can try and deal with it on a more strategic end to try and improve our endpoints and our precision in our analyses to make that 20 to 30,000 patients into 5,000 patients across a variety of indications. And again, that requires this intersection of understanding the physiology and the science to not only understand the basis for the disease, but then come up with better biomarkers. If you look across the breadth of the heart failure space right now, we really don't have any very good biomarkers. We can use things like NT-proBNP, but we could debate in different forms of heart failure what our level of confidence is in that biomarker to give us a hard endpoint, and certainly as a registrable endpoint, that's not very practical. So how do we support those biomarkers? How do we find those new biomarkers? And in doing that, how do we match them with the right patients? I think that those two sets of strategies exist. We have to find a better way to do this because, as I said, following 20 to 30,000 patients for up to 5 years apiece is not a way to evaluate multiple therapies.
I think that it also involves working with the regulatory agencies to understand how the burden of heart failure is not just about the mortality, which is certainly severe, but about the morbidity and the limitation on daily activities of many of our heart failure patients, particularly the inherited cardiomyopathies. I'm also thinking of the African Americans with the V122I transthyretin amyloid mutation, who are limited in their activity. They're easily short of breath. They're not able to work to their full potential. They're not able to care for their families for the full potential. And understanding how endpoints such as submaximal exercise can be quantitated or maximal exercise can be quantitated in a way that I think would meet objective criteria for approval of a new therapeutic, I think is something that I think about a lot.
So I get paid to run big clinical trials for the company, so I'm happy if we still have to keep running them. I'd like to stay in this job for a bit. But, having said, think back, I think it was J.B.S. Haldane, the Nobel Prize-winning British physicist, who at one point said, if you need statistics, you've designed the wrong experiment. At a certain point, if we understand what's going on enough, the effect size from our interventions is big enough that you don't need 20,000 patients in a clinical trial. Right? The reason that we don't need randomized clinical trials to prove that you ought to put on a parachute if you're jumping out of an airplane is because we understand what's going to happen if you don't, right? We're not at that point, and we're not going to be at that point in heart failure for a very long time. So what happens in the interim? I think what happens in the interim is as we better subgroup patients, we're still going to be left with large populations. But hopefully, the effect sizes of our interventions get to be so much bigger, right, that we don't need to do 20,000 patient clinical trials. And the other point I think we need to make in terms of thinking about clinical trials is that we also need to run clinical trials of strategies rather than clinical trials of single interventions. a lot of the ways that we're going to have to deal with heart failure will involve combinations of therapies. And, I've posited to you, and I think, everyone here has said the same thing, that we want to get to a world where we identify a patient's specific physiology and where we treat based on that. But, that's an assumption right now. That needs to be proven. And that is the sort of thing that we can prove running pragmatic clinical trials to identify which strategies lead to the best outcomes for patients.
But can we get a little bit better about picking winners earlier, right? Do you think we can do a better job of anticipating rather than waiting the full 5 years before the trial is done, hundreds of millions of dollars in some cases invested in the experiment? What can we do to learn a little bit earlier on which patients are benefiting from technologies that we're deploying, which patients are likely to be the ones we should be recruiting? Are there reliable surrogates kind of efficacy that are on the horizon?
So it's a challenge, and this is what I do. I don't run the large clinical trials. I run the phase 2 studies to give confidence in in the large trials, and that's the challenge we face, is how to de-risk those trials. This is still a very difficult space to do that in. We can use certain serum biomarkers. We can do functional tests with 6-minute walk tests and things like that. We can get some capacity to do this. But I will say that it's still a big bet in our best day. As we refine the patient population, the magnitude of effect, as Ken says, gets bigger, and we have a better discriminatory capacity to predict those trials, which, yes, then will lead to regulatory approval, payer approval, and all those important pieces. So we're trying to do that, but I think it has to be coupled with new science. I don't think that the biomarkers we have available to us today, even if we combine standard serum biomarkers with cardiopulmonary exercise testing, with electrophysiologic testing to understand the adjacencies, the heart failure, et cetera, I don't think they're sufficient to do that.
But let me defend, Craig and the translational biologists for a second, right? Because, again, a lot of the pessimism, I think particularly in the drug world about heart failure therapeutics, is that there's just been this whole series of agents that looked great in early trials and then never translated or didn't translate successfully into the phase 3 pivotals. And you can ask, is that because the translational science, the basic science is wrong, which I actually think is pretty unlikely. Or it gets back, or are we just trying to deploy the drugs in the wrong way with this kind of one-size-fits-all strategy? I mean, you look, again, coming back to the late-breaking science from Paris this week, 2 agents that have been very promising in early trials, ularitide and serelaxin, both failed to meet their endpoints in large pivotal trials. And I think you have to ask yourself, is that, again, because the drugs aren't going to be effective in heart failure patients, or is it because we just haven't figured out when to give them, how to dose them? Should we be giving them earlier in the course of disease rather than waiting for someone to actually present in the emergency room with their heart failure decompensation? And so, again, I would surmise that a lot of agents that we've sort of thrown away because they, quote unquote, failed in their pivotal trials would have done much better if we had the opportunity to match them better to an understanding of a patient's underlying genomics, proteomics, and current physiologic state.
I would add that the TopCat studies of spironolactone, which were successful, I believe, in the Western Hemisphere. Actually, I think
Thank you for saying that. Yeah.
were very involved in that, and overall had a null result because of perhaps a different type of patient that was addressed in Eastern Europe. But I also don't want to have us forget the importance of preclinical models that are more and more informed by what we're seeing in the clinic. And whether our ability to now create genotypes, even in larger animals, where we can assess hemodynamic changes and biomarker changes, and even imaging changes can allow us early insights into some of the different phenotypes of heart failure.
You know, I think someone earlier mentioned that oncologists have been doing this kind of work for years and are a little ahead of us cardiologists. And the key to the oncology model really is to view translation not as a one-way street. It's not from bench to bedside. It's bench to bedside and bedside back to bench again, right? And we've been less effective than that historically in the cardiovascular field. But that's really the key here, is to have those animal models, to have the early clinical studies work together, then to do a large trial, and then to begin to understand from ularitide and serelaxin what was the physiology that was seen in those trials. How do we take that back and revise these drugs, revise the populations, and then re-explore again? Of course, we need to have better markers along the way because we can't keep running. I can assure you from our end, I'm not going to get a lot of support to keep running lots of RELAX-2 trials. But we can do this. We just have to keep going back and forth and reiterating this process and finding those newer markers from the clinical data sets we had to inform the next trial.
And I think to circle back to your, I think one of the intents of your question, I'd like to take an opportunity to publicly, as an industry representative, thank the FDA and actually applaud them for the expedited review pathway process, in which 3 essentially non-hard outcomes, if congruent, being biomarkers, functional status, and quality of life. If all of those are positive, then the approval pathway can begin, and adaptive Bayesian statistical methods and designs of trials can move on from the regulatory pathway into an earlier market development. I think that's been a major step forward to identify earlier when things seem to work and seem to win or not seem to win, so that you don't spend all of the time before you look to see if it worked. And I think that sort of kludgy old way of approaching clinical trials has to change. And I do believe with the availability of big data, with all the caveats that's already been mentioned, I'm reminded of a patient that I heard about who was in a trial with a wearable, which I'm a big fan of, but found out that he put it on the washing machine while it was running so that he could tell everybody that he was actually active that day. So, you know, I think
In our trials, they usually put it on the dog.
So, you know, garbage in, garbage out is absolutely true, and we run the risk of having such big databases that we actually just simply create questions that are not clinical needs. And so there's a lot of caveats there, but I think becoming more creative in how we do randomized trials, I believe, can allow us the opportunity to identify what's good and what's not earlier in the process. As long as there's congruence with the market development and reimbursement and coverage and all the things that go along with, oh, so you have FDA approval, so what? I think that congruence is in evolution, and I see that as being successful over time, but certainly one of the things we're wrestling with now.
So it's amazing how quickly this time passes, but these are all big topics, and I guess as we take the last few minutes here, one of the things that I wanted to do was to walk down the line. Maybe we'll start with you, Ken, but really to just ask you, given the challenges you've laid out, the difficulty in phenotyping populations, the difficulty in harnessing the value of big data informatics to define clear, discrete entities within this complex problem, the regulatory challenges and some of the challenges in implementation that you mentioned, what keeps you engaged in this space? You represent companies who've made a commitment to heart failure individually, and we applaud that. And I guess the question is, what is it that keeps you here, and what is it that makes you optimistic that we can solve some of these very big problems that you've laid out? So maybe, Ken, from your end.
Yeah, I mean, why be optimistic? Well, I think because if you weren't optimistic, you'd kill yourself. And it comes back to where you started. There's just such a tremendous need for our patients. And again, you know, those of us who are here are physicians, nurses, nurse practitioners who've taken care of these patients, I think really understand just how badly they need us to improve their care. So there's that. Is it difficult? Well, yeah, but if it was easy, we'd have done it already. You know, the opportunity, frankly, from an industry point of view, only comes in if things are difficult. That's why we think we're going to be the ones to do it ahead of everyone else. I mentioned, again, our multi-parameter heart failure diagnostic. That program was 15 years in execution, now leading up to anticipated FDA approval. You've got to be in this for the long haul if you want to get forward with it. But if you are in it for the long haul, I believe, right, and that the example from other things like oncology, or even coming back to something simple, endocrinology and diabetes, right, says that there is a model that will be successful if we work hard enough at it.
Akshay, you and I know that there's a huge unmet need. I mean, I'm still close enough to the advanced heart failure practice that I know that transplant is the solution for very much a minority of patients, and that you're trading one set of problems for another. You know, mechanical circulatory support, certainly an important advance, but again, of limited utility, including to those large number of patients that are quite limited and remain, you know, the major cause of rehospitalizations, the heart failure preserved ejection fraction group. I mean, we've got to do a better job with both the younger patients who perhaps more clearly have an inherited basis to their cardiomyopathy, and the older patients in whom I think there's a mix of acquired and inherited factors that lead them to be basically, you know, ridden with the burden of heart failure.
So, Akshay, I mean, why do we do any of the science that we do? From the times, you know, I was a basic scientist early in my career to the work I do now, it's about having the tools at hand to answer questions that excite us, that we think are going to be impactful, and then every once in a while having a little success to tease us along to the next opportunity. Since the days that, at our company, since the days that Marc Fishman, someone I think known well to this audience, initiated the Novartis Institutes of Biomedical Research, there's been a very simple algorithm to how we decide what projects are or where we focus. It's a 2x2 grid with an axis, an X and a Y axis. With on one axis, we have unmet medical need, and on the other axis, we have new science. And there's plenty of that in heart failure. There's plenty of unmet medical need, and there's plenty of new science. So the tools are there, and it's very exciting. It's an exciting space to work in, and we know we're doing something important. And every once in a while, in all of the work we do, we get a little bit of excitement and a little bit of success to drag us along to the next opportunity. In our team, my team at Novartis was one of the teams to early invent Entresto. My team was very excited to see those paradigm results, right? And to see a 20% reduction in heart failure where we hadn't had a drug to do that in many, many years, as you all know. And so that reinvigorates you, right? And it moves you to the next opportunity. And along the way, not only do we see a clinical trial results, but we get word back from patients, friends, families, the patient who takes Entresto, who we hear the stories a week or two later, come back and say how much better they feel. It's that inspiration from our patients, along with the excitement of doing the work, I think, that makes us optimistic to move to the next program to keep going.
Well, you know, my why is very similar to the rest of the panel's why, and that is this tremendous potential to actually change the course of a person's life who has what has for many years to be thought to be a invariably fatal and horrible disease. I think that we have so many things that are coming to the forefront that makes the hope for the patient with heart failure gleam in my eye, having looked in a lot of eyes that have the syndrome itself. I mean, it's kind of like, why do you play golf? You know, why would you even do that activity? It's that birdie you make in hole 17 that brings you back to play again. I think that we keep persisting, as Ken mentioned, 15 years, same thing with hemodynamic monitoring. Put the first sensor in 1997, and you know, 2014 is the first system approved by the FDA. So there's a glory of being persistent and seeing that work, the hard work, pay off, as long as you can live and survive long enough to see it.
Well, I love the golf analogy, and I'll just have to take your word that people make birdies from time to time. But I commend you all for your commitment to the space. And as a practicing heart failure clinician, I'd say that I'm really hopeful that you maintain that commitment over time, because we certainly need more therapies, and we need to apply the therapies we have more effectively. And I think a lot of this opportunity for continued collaboration between us and the academic world and you on the side of industry, I think is really is part of that path forward. And hopefully this collaboration can also feed some innovation within the regulatory environment that can hopefully smooth the way and short circuit the development of new therapies and their application in practice. So I want to take this moment, because we're at the end of our time, to thank all the panelists for participating in this discussion. I hope you found it useful, and I think we'll close here right at the second on time.
Thank you.